Okay, so thank you all for coming. I'm Claudia, and I'm going to present some preliminary results of my thesis, because I still haven't finished, on the topic of global patterns of seaweed species diversity. My thesis was supervised by Christophe and Quinten from the Phycology Lab in the UGent. So, it's going really slow. So probably you're asking whether this was already studied. And yes, we have been studying biodiversity patterns for decades, but in fact, in recent years, due to an increase in anthropogenic pressure, there is a need for understanding how these patterns work and the underlying processes that might generate them. And this has been resulted in an increase in the number of papers related to this topic. For the marine environment, the focus has mainly been on animals, with really few papers related to primary producers. Specifically for macroalgae, that is my topic, there is a lack of studies at a global scale, and the few that are present, they are developed at the level of genus, like this one from Kerswell. So, however, increasing the taxonomic resolution may produce different patterns. When studying biodiversity, it is important to account for latitudinal patterns of biodiversity. For the marine environment, it has been studied for also decades, and in this review by Chaudhry, he showed that in most cases, the pattern observed is bimodal, so meaning that higher richness is observed at temperate regions. And finally, related to biodiversity, it is also important to create a compartmentalization of biodiversity, and thus it's the generation of biogeographical realms. For example, in the upper figure, I have geographical realms for the terrestrial environment. 2 years ago, Costello proposed 30 biogeographical realms for the marine environment, but the marine flora were not included. With this in mind, I'm going to present you my goals of my thesis, and it's first to assess the data completeness of databases, of the databases that I'm going to use, to then study the diversity patterns per se, and to investigate which environmental variables could explain the patterns observed, and to finally create biogeographical realms specifically for macroalgae. So how did we do it? We first got the data. We'd use georeference occurrence data from OBIS for the 3 main groups of macroalgae, and we submitted this data to a data cleaning process to only keep the correct names and correct coordinates. We also downloaded data from AlgaeBase, but that is a repository of algae species. But there is a huge difference between the data provided from OBIS and the one provided from AlgaeBase. For AlgaeBase, there is only presence data, and in fact, it's just a list of localities where the species have been recorded. So this gives us 412 localities that we manually georeference. But because of the nature of these localities, we have whole continents like Antarctica or islands or countries. This gave us 67 problematic regions that we created a Python script that was able to, for these ecoregions to to include the ecoregions present for these different localities. And the ecoregions are the base of our study, and they were developed by Spalding in 2007, and it consists in a nested system of 12 realms that are the higher spatial resolution, and then province, and then ecoregions. In this map, you can see the ecoregions in white, and I'm sorry that you cannot see the other ones, but I have a problem when I exported my PowerPoint as a PDF, so only the first image was recorded. So for OBIS, we calculated the number of species per each of the ecoregions. And as we can see in this map, where I have all of the occurrences for the 3 groups, there is the sampling effort is not even in the globe. So in order to correct for sampling effort, we tested 2 different approaches. One is a curve fitting approach, where we fitted models to the species accumulation curves per each ecoregion, and we chose models that show best performance in the literature, and we did it with these 2 packages. We also tested non-parametric estimators that they account for the undetected species in our data by using the rare species. And we choose these 3 that are the recommended for incidence data. And completeness was then assessed as the proportion between the observed and expected species. This is, Okay, so this is a map showing the completeness calculated by the KO estimator. As you can see, there is a higher number of ecoregions that show completeness, but still for OBIS, there are quite a number of ecoregions that have a completeness of less than 15 species. So to further investigate how well is the OBIS data, we compare the species, the number of species for AlgaeBase, for AlgaeBase and for OBIS. And in this graph, you can see the log of the number of species and the share and unique species for each database. So in all cases, AlgaeBase show a higher number of species and a higher unique number of species. And the fact that we encounter unique species for OBIS is like problematic because we shouldn't have them if we don't have them in AlgaeBase. So this is another reason to don't trust that much OBIS. And this is why for the rest of my presentation, I'm going to show you results on AlgaeBase. So again, for AlgaeBase, we calculated the number of species per ecoregion, province, and realm. And for the latitudinal gradient, we created 5 degrees intervals and accounted for the present absence of the species in each interval. And to detect undetected species, we define a missing species as the one that is not present at 5 degrees interval bands from the upper to the lower limit of a species. So imagine that we have a laminaria at 20 degrees and at 10 degrees. The fact that we don't find it at 15 degrees is considered a missing occurrence and is added to the missing plot. So yes, these are the processes. So these are the maps that we observe at the level of ecoregion. The patterns are not clear. More or less, we can see that the temperate regions have a lower number of species, and the Mediterranean and the Caribbean and Southeast Asia show higher number of species. But when we decrease the spatial resolution to the level of province, Okay, it's going really slow, okay. So when we decrease the spatial resolution, we see that the richness is shared between the Mediterranean, the Madagascar, and the Caribbean, with the highest number of species still in the Pacific coast of the United States. And is it only at the level of realm, okay, when we see a similar pattern at those observed at genus level, with a higher diversity present in the Pacific Ocean, and the highest diversity of all present in the temperate regions of the Pacific Ocean, such as Japan and the East Coast. And for the latitudinal gradient, we did not observe the same pattern of marine environment. In this graph, we have the missing species in gray, the observed in blue, and the corrected in red. That is just the sum between the missing and the observed. So as we can see, there's still some dips in my data. That is because I don't have it completely yet. But we can see there is a higher richness in the tropics for macroalgae. And this pattern is repeated for each of the groups. So for the green, the brown, and the red. So to investigate what can cause this richness, we downloaded potential predictors of macroalgae richness related to temperature, oxygen, and nutrients. This is an example with the sea surface temperature, and we calculated the mean per each variable inside each ecoregion. Before applying models, we tested for correlation, and we finally only got 5 environmental variables that are able to be used in our analysis. So first, we generated ordinary least square, that is a type of linear regression, to test for assumptions of linear regression and also to test for a spatial autocorrelation of our residuals. we did find a spatial autocorrelation, and to account for this, we then generated generalized least squares that are able to cope with a spatial correlation in the residuals. We also did a geographical weighted regression to calculate whether there are some areas in the globe that have more explanatory power than in other areas. But this is still in the making. So I can show you the generalized least squares, and we observe that variables that explain the richness are related to temperature and to nutrients concentration. And this has been repeatedly observed in the literature. And in fact, there are several hypotheses that explain richness in tropics. And one is related with temperature, assuming that higher temperatures provoke a higher metabolic rate, and this favors speciation. And these are the fitting of my models. They are quite low. One, because I'm doing it at a global scale, and second, because maybe I did not account for variables that are important predictors. And finally, I did the biogeographical realms using clustering algorithm. I tested 7 different clusters, and the best one was Ward. And the recommended number of clusters was 9. So to show this better in a map, I plot them here where colors indicate similarity. So there is a good compartmentalization of biodiversity, and we could really easily differentiate different realms. But there are still some missing data. So to conclude, so far, what I've discovered is that the OBIS is exhaustive, but taxonomically not sufficient for this type of analysis, that seaweed biodiversity pattern at lower level scale differ from those found at genus, and then latitudinal diversity for macroalgae follows the trend found in terrestrial ecosystems, and this can be explained to almost some extent by temperature and phosphate, and there is a good distinction of biogeographical realms from macroalgae. So the further research that I still have to do is to first complete the AlgaeBase data, as the addition of missing regions might show different patterns, and to try to include variables such as substrate availability or depth in the models that could improve the fitness of my models. And then to investigate whether same variables explain species richness at different taxonomic levels and at different macroalgae clades. Because, for example, the Dales shows a more tropical pattern than laminariales. And in the end, what I would like to do, but I don't think that I will have the time, is to investigate what are the variables that explain the current barriers that we observe in my biogeographical realms, whether this could be caused by tectonic movements or by climate heterogeneity. So these are my references, and thank you very much for your attention.